Welcome to Omics Revolution Unpacked, where we explore how large-scale biological data is reshaping personalized medicine. In this presentation, we'll map the key omics layers, highlight crucial technological and computational shifts, and explain how integrating genomics, transcriptomics, proteomics, and metabolomics is opening new routes to precision diagnostics and treatments. Genomic and other omics data have exploded into petabyte-scale archives, driven by rapidly falling sequencing costs and new infrastructure needs. Public and private repositories now hold enormous volumes, forcing a shift to cloud storage, federated analysis, and scalable compute, while bioinformatics evolves from legacy lab workflows to AI-integrated, real-time analytics. Variant analysis begins with careful reference assembly and consensus calling, then moves through pipelines that detect SNVs, indels, structural rearrangements, and copy number changes. Population allele spectra and global cohorts help prioritize rare versus common variants, and annotations tie genotype to phenotype, using functional impact, evolutionary conservation, and clinical databases for integrated interpretation. Epigenomics uncovers how the same DNA sequence produces cell type-specific expression through DNA methylation, histone modifications, and chromatin accessibility. These assays define enhancers, silencers, and topological domains that shape genome organization, explaining why two cell types with identical genotypes can show completely different gene expression patterns and biological behaviors. Transcriptomics captures RNA-level activity from bulk RNA-seq to single-cell and spatial methods, quantifying mRNA, non-coding RNAs, and splice isoforms. Single-cell and spatial approaches resolve cellular heterogeneity and map expression in tissue context, while downstream analyses translate these profiles into differential expression patterns, pathway enrichment, and mechanistic hypotheses for biomarker discovery. Proteomics uses bottom-up LC-MS workflows to identify peptides after enzymatic digestion, then quantifies proteins using label-free or isobaric strategies and advanced approaches like DIA and PTM mapping. Importantly, protein abundance often poorly correlates with mRNA levels. So proteomics reveals translational control, degradation dynamics, and functional context that RNA alone cannot provide. Untargeted metabolomics combines LC-MS, GC-MS, and NMR platforms to detect diverse metabolites, followed by raw data acquisition, peak alignment, spectral library matching, and pathway topology analysis. This workflow bridges genotype and phenotype by providing real-time physiological readouts, useful for nutrition studies, toxicity assessment, and early disease monitoring through metabolic signatures. Microbiome sequencing ranges from 16S rRNA amplicon profiling for taxonomic composition to shotgun metagenomics for functional potential, revealing strain-level variation, horizontal gene transfer, and metabolite crosstalk with the host. Perturbations in microbial communities, or dysbiosis, are increasingly linked to inflammation, metabolic disease, and neurodegeneration via gut-brain signaling pathways. Integrating multiomics relies on core frameworks like correlation analysis, Bayesian inference, and machine learning, plus advanced models such as multi-block PCA, network propagation, and pathway-centric approaches. Critical prerequisites include data scaling, batch effect correction, and missing value imputation, because harmonized, high-quality inputs are essential for meaningful biological interpretation. Predictive modeling in omics uses genome-scale metabolic reconstructions, kinetic ODE systems, and context-specific flux balance analyses to simulate biology and predict outcomes. These models support growth predictions, drug target identification, and biomarker secretion, and they improve through an iterative cycle of new experiments, model refinement, and validation to boost predictive accuracy. Translating omics from sample to clinical action requires robust workflows, starting with standardized sample collection and biobanking, then multi-omics profiling and computational interpretation. Outputs feed into clinical decision support systems, trial matching, and physician guidance, while ethical safeguards, physician education, and equitable access are emphasized to ensure responsible and inclusive deployment. Looking ahead, technological forecasts include real-time wearable metabolite sensors for continuous monitoring, single-cell multi-omics atlases for high-resolution cellular maps, AI-driven digital twins for whole-body molecular modeling, and quantum-enhanced analytics for complex pattern discovery, all enabled by ethical data sharing, interoperability, and global equity to maximize societal benefit.